Hello, everybody. It's my great pleasure to welcome you to Bachem's webinar, the use of peptides in the production of peptide active pharmaceutical ingredients. As a short notice at the beginning, I want to say this webinar has been pre-recorded, but the Q&A session will be a live event. To the speakers of today's webinars, I'm happy to introduce Leendert van den Bos. He is CEO and co-founder of EnzyTag, and Robert Naas, who is business development consultant to EnzyTag. As you might know, we have a cooperation, Bachem has a cooperation with EnzyTag on adapting the CEPS technology for the manufacturing of non-GMP and GMP grade peptides. My name is Bernd Raschak. I'm a business development manager here at Bachem, and I will be the moderator of today's webinar. Before we go into detail about the EnzyTag technology, I will give you a short overview of Bachem first, and then hand over to Leendert. So, Bachem, what we are doing is that we are a pure contract development and manufacturing organization, meaning we are producing drug substances or APIs. It's worth to mention we don't have our own drug development. We don't have our own drug product pipeline. We have just celebrated our 50th anniversary last year, and we are proud to still be independent now for over 50 years in the field. Our main focus is the production of GMP-grade peptides, and that for decades we have been the market leader in the place. And for several years now, we also expand our pipeline or our manufacturing to oligonucleotides, also on a GMP manufacturing scale. We have about 170 different drug development projects where we are involved with our partners, which are mainly big pharma companies and biotech companies. And by contributing to these organizations, we also play an important role in supplying beneficial drugs to the society. Okay, on the next slide, you will see Bachem, we have given us 5 strategic foundations in order to achieve our goals. This webinar mainly focuses on the innovation and the sustainability part. I will show you what we do for our customers. So we support our customers from research to commercial API supply. It typically starts when you do a drug development project at a university or biotech or also pharma company with a lot of lead candidates that you need in small non-GMP quantities that can be supplied from Bachem at inexpensive prices. When you have selected your lead candidate, then it goes into toxicology studies. We can deliver the non-GMP tox batch as well as the following GMP batches, where we do an optimization during development when our customers are in phase 1, 2, and 3. And then hopefully we have successfully concluded a process validation step. And our RA has compiled a drug master file so that we can give our customers these, that they apply for this new drug and hopefully get an approval. And if this drug is approved, Bachem's strategy is then to supply our customers on a commercial scale. And even if the substance patent is expired, we can further supply the NCEs. Since the drug development is a quite complex issue, we have a dual approach to cover that with the business development and the project management. The business development is responsible for commercial issues, whereas the project management is taking care about technical aspects of these drug development projects. On the next slide, you can see we have experienced, especially during the last decade, that there is an increasing number of types projects, meaning oligonucleotides and peptides projects, as well as the volume per API project is also increasing. So there's a clear market demand for capacity and capability expansion. And our board has decided to make a capital increase last year. We have secured about 570 million Swiss francs, which we already put in existing buildings for capability and capacity expansion. But most of this money will go into the new tides manufacturing building, which will be operative in 2024. I will show this in the next slide soon. And what I still wanted to say is, because there's such a big demand, especially also for oligonucleotides in the last several years, that we have decided to really implement a total new manufacturing site in Switzerland that will be most likely in the northwestern part of Switzerland. This site has an own QA, maintenance and storage, and this will become hopefully active, operative at the end of this decade. On the next slide, I show you the building K, which is currently in construction. This is maybe the world's most modern tides fabrication plant building. When we have built it, there are 2 phases. After the first commissioning, we have more or less already doubled our existing capacity here, what we have here in Bubendorf. And when it's fully operative, we will create approximately 300 new jobs in this region, which is really great. As mentioned, from this 570 million alone, 350 million will go into this building. So this is really a huge amount. What we do here is also to adapt the green chemistry approach. For example, we implement new technology for wastewater treatment, and we try really to reduce solvents and as many toxic chemicals as possible. What we do here as well is also the adaptation of the Industry 4.0 dogma or strategy, meaning an increase of automation, a real-time data exchange of manufacturing processes, as well as implementing a paperless lab environment. So by use also of digital batch records. Yes, and this shows that we are really doing a lot of innovation within Bachem. But of course, and that is shown on the next slide, we are also very open to cooperate and drive innovation with external partners. And yes, and this webinar is about our collaboration with this young and technology-centric company EnzyTag, which is a perfect example for a combination of innovation and greener chemistry. With that technology, we can really produce long and cyclic peptides in a very elegant way. This is already the 4th webinar on this SAPS technology, and I'm now very happy to hand over to Leonard, who will give you a deeper insight. I say thank you and forward to Leonard. Leonard, the show is yours. Thanks.
Thanks a lot, Bernd. Hello, everyone. It's a great pleasure to be here again and present to you in our chemo-enzymatic peptide synthesis series more on what we're doing. So the next slide shows you a bit of the work or the agenda that we would like to cover in this webinar. We would like to start off with, again, a short introduction on the chemo-enzymatic peptide synthesis, so just a fresh up that we're again on the same page. Then we will present an historical perspective and recent examples on the use of enzymes in API production. And the last topic we would like to cover in this webinar is the quality and regulatory aspects of it. So I will present the first 2 bullets, and my colleague Robert Naas will present the last bullet. So again, as a quick recap of what the peptide ligase technology platform exactly is and what EnzyTag is doing. It's an enzyme platform for the assembly of peptide-containing therapeutics and bioconjugates. It has been proven already on various peptide APIs. Some of them are in the generic field, and others are in the NCE field. It is also an enabling tool for complex cyclic peptides and small proteins. So where you couple multiple fragments together, or where you cyclize certain peptides, long or small peptides. Because of the relatively small recognition sequence in the enzyme, you can also use it as a ligation tool for connecting linker systems and non-natural fragments in the bioconjugates, biospecifics, and ADC space. EnzyTag is a small company. We have no production facilities. We only have lab facilities. And that's why we're very happy that we've entered into a technology transfer and license agreement at Bachem that takes care of the cGMP and process development aspects of the SEPS technology. On the next slide, you see how the SEPS technology exactly works. So we've presented two examples. At the very left of the slide, you see how a linear peptide is assembled from two fragments. And in the middle, you see how the cyclization works. So you have here fragment 1, which has at its C-terminal a leaving group, an oxoester. And at the N-terminal, it has a suitable protection group. So we have a second fragment that would ligate to the first fragment. And then by means of the peptide ligase recognizes this green hexagonal picture over here, which is the oxoester. And the cysteine of the peptide ligase enzyme reacts with the oxoester, forming a thioacyl complex. Then fragment 1 is covalently attached to the peptide ligase enzyme. And in the second step, fragment 2 comes in and gives the actual ligation. So the actual amide bond forming step takes place in the second step. And after that, we remove the protection group at the N-terminus of fragment 1, and that gives you the new fragment, or it gives you your product, of course, if it only exists of two fragments. And the new fragment could enter into a new cycle, which can be started. In the middle, you see how the cyclization works. There you have the very same starting position, but now you lack the protective group at the N-terminus. Here you have the leaving group, so the oxoester. The peptide ligase recognizes the oxoester, and then the tail of the peptide backbone attacks to the head, so to the activated C-terminus, forming an amide bond and essentially then giving a cyclic product. We say that the technology is footprint-free, which means that the peptide ligase enzyme does not recognize a single sequence of amino acids. So multiple amino acids are tolerated in the recognition pockets of the enzyme. You do not have complete freedom, so there are certain rules and certain limitations, but it is so on average some 10 to even all 20 amino acids are tolerated in the various pockets, and we can provide you more info on that separately. It's very efficient. So in general, you need only grams amounts of enzyme to create kilograms of API. And of course, it goes without saying, enzymes and aqueous environment is very green and very sustainable. The fields we see applications for this technology are peptides, or long peptides, typically the longer than 60 amino acids. That's a bit, of course, sequence dependent, but so the ones that are outside the reach of standard solid phase synthesis. All peptide cyclization, longer than 25 amino acids. Also, this is a bit arbitrarily, of course, because it strongly depends on the sequence. And in case you would like to have functionalized proteins, so that are like 100 mer or 120 mer amino acid protein that is functionalized in the protein backbone at its lysines, for example, with a PEG or with some other modality, and that can all be synthesized with our technology. And the benefits, it's scalable under cGMP, that has already been proven on various APIs. no side reaction, so it's extremely specific to the termini of the fragments you would like to couple. It's organic solvent free, so but on the other hand, you can add a bit of organic solvent in case you do have solubility problems with your fragments. You can also add chaotropic agents, and unlike native chemical ligation, you need no cysteine nor subsequent desulfurization step in order to remove the thiol side chain in order to ligate peptide fragments together. Oops, a bit too fast. So here is again a picture that I took from the ACS Green Chemistry Roundtable, where they plotted the sustainable aspirations in the field. So let me start with the gray classical strategy with solid phase peptide synthesis, which is typically linear synthesis, making use of heavy toxic solvents. You need a lot of protection groups, intensive chromatography at the end to purify, so mainly requiring loads and loads of acetonitrile, and you have a very high process mass index. Then the sustainable aspirations are convergent hybrid synthesis, so making use of multiple fragments and ligating them together in one way or the other. Greener solvent choices like methyl THF and NMP. Unprotected peptide ligation, which does not require the removal of an enormous amount of protective groups. Greener separation technologies and innovation to reduce the PMI. And here we've just positioned SEPs in the middle. We still make use of solid phase peptide synthesis to make shorter fragments, not the very long fragment or not the very long API, but the shorter fragments, and then ligate them together in buffered aqueous conditions. The enzyme does not need protected peptides, so we can work with completely unprotected peptides, ligate them together, and essentially giving you your product in one step. And because the purity of the shorter fragments is higher, we selectively ligate with our enzyme the fragments of interest together. The overall purity of the product is higher, which results in less solvent use during the purification. And on average, a 90% reduction in key PMI parameters is reached, and that can be read in an article published by EnzyTag, amongst others, in the Green Chemistry Journal. And here you have the digital object identifier referring to that article. So that was just a short recap of SEPs and also an outlook towards the green and sustainable benefits of the technology. I would like to continue with an historical perspective and some recent examples on the use of enzymes in API production. And here you have a timeline which shows which puts a bit of historical perspective to what we are discussing. And it goes back to the late 80s, the early 90s, where the FDA was mandating the removal of isomeric ballast in APIs. So, of course, everybody remembers the thalidomide story, and the FDA wanted to overcome that, and they demanded that only single isomers should be marketed as drugs and no longer mixtures of isomers, which cause, of course, side effects. So a second very important thing was the US Pollution and Prevention Act in the 1990s, which focuses on waste prevention instead of end-of-pipe waste treatment. So companies in general should care about the kilograms of waste they generate rather than take care of a very good waste treatment in the end. So in case it's not there, you don't have to treat it. So for that, a factor was defined called the E factor, which says how much kilogram of waste is produced per kilogram of product. That's not only applicable to the API field, but it can also be used to other chemical industries or to other industries in general, to other products. But it was also adopted in the pharma industry, and by then also a first example of what is possible with enzymes was published in the penicillin field. So where penicillin G acylase was used for converting penicillin into novel or newer antibiotics, and more on that on the next slide. And the whole field of the enzymes, enzyme engineering, enzyme optimization was boosted by the surge in molecular biology and the whole DNA revolution we all have heard about. So now we have many biotech companies that play around with the genes of microorganisms or other organisms that optimize enzymes. So we have now computer algorithms that could predict what would happen if certain modifications are applied to enzymes. And now, very recently, we have also the artificial intelligence, which is sort of with even bigger steps forward, predicting what would happen in case some 10% or so of the enzymes changed for other amino acids. So an example that I already touched upon was the conversion of penicillin G into, in this case, ampicillin is presented. Back in the days, before Pen-G amidase, before the enzyme was invented, it was done with dirty chemical steps requiring multiple steps. And then scientists discovered that Pen-G amidase could essentially do it in one step. So convert penicillin G into 6-APA, into the 6-APA intermediate. And this intermediate was then the starting material for creating a whole range of these type of antibiotics, where different groups were attached. So in this case, it was this amide that was coupled with another variant of Pen-G amidase, which was also engineered in a way that it could couple 6-APA to this intermediate, giving ampicillin. And that could not have been realized without the recombinant production of Pen-G amidase in large quantities in factories. Further, was already invented back then that enzyme immobilization was very crucial to bring down the cost. The second example is sitagliptin. I think this is really a landmark example where Codexis and Merck Sharp and all partnered together in order to introduce several enzymes for several production steps. And I just want to highlight the one that is placed in the box. That's the conversion of this hydroxyl, so chlorohydroxy compound via halohydrin dehalogenase into this cyano compound. And so this table shows you the improvement that they were able to realize, where several parameters were investigated. So the concentration of the substrate, the concentration of the enzyme, the catalyst productivity, also space-time yield, which is a measure for how much catalyst is required to convert a certain reactor volume in a certain time period into to convert that into product, and enantiomeric access. And here you have the wild type, so the starting enzyme that was 20 gram per liter, for example, for the concentration of the substrate, and the best variant in the end called 140. And also the concentration of the enzyme, so was brought down significantly from 30 to 1.2. Catalyst productivity was boosted enormously. Space-time yield, so they could create way more product in the same reactor volume. Anti-enantiomeric access was high and still is very high. So they were able to conserve that in the enzyme halohydrin dehalogenase. And this is not the only, so this graph shows you a bit of the work and also more recent examples of where the world is working on. So in this graph shows you how many enzymes are involved in cascade reactions. So where multiple enzymes are used in one pot to convert a certain substrate into product. So typically we think of, well, one enzyme per one conversion, then do a workup. But you could also just, because of their selectivity and specificity, you could also add multiple enzymes together and then let them interact with the intermediates and the intermediate compounds that are formed, and take it one step further and even further reducing the production burden. Atorvastatin from Pfizer is then one example, which was published in 2019, which makes use of 3 enzymes. And islatravir from Merck Sharpendome, again, they are very active in this field. They use quite a bit of enzymes, so just a bit less than 10, and that was published in 2019. And here you have the latest example, which is molnupiravir, which is the COVID drug that they got approved in 2021. I believe this was also in collaboration with Codexis, where they used multiple enzymes to produce this complex drug. And so I think we're just at the start. So many pharma companies, many biotech companies are investing in this. And the de novo enzyme design will further boost the field. So I think over the next couple of years, we will see many, many more examples of the use of enzymes in peptide or in API production in general. And then over to Robert to hear more about the quality and regulatory aspects.
Yes, thank you, Leonard. So as Leonard explained, and Leonard has given some examples of the use of enzymes for API production. And this is now quite common and increasing, as he mentioned. But we still, when we talk to customers that are interested potentially in applying our technology for the production of their peptide NCEs, we still get a lot of questions regarding quality and regulatory aspects of the use of enzymes. So what I want to do in the next part of this webinar is address some of these questions, both in a general way, and also focus a little bit more on the peptide ligases and how to look at control strategies regarding peptide ligases. Okay, so as an example of some of the questions regarding the use of enzymes in a GMP environment that we get, we've listed some here. This list is by no means extensive and complete, but I think these are some of the more interesting questions. So like, what are the risks associated with the use of enzymes? Very important is how can you remove an enzyme from your product, or an enzyme related impurity from your product? And how can you analyze and detect that and show that you have removed that? Of course, very important is how to clean the equipment when you have used an enzyme for production. And also very interesting and important is, can you use or can you optimize an enzyme? And when in the clinical development is that still allowed? So again, I will address all these questions in a general way, and also focus on peptide ligases. So before going into these questions and the answers very specifically, a very important question to first ask is which rules are applying here. And if you read the ICH guidelines, you will not find a section or part of the regulation that specifically talks about the use of enzymes for API production. So does this mean that everything is allowed? Well, no, of course not. That's not the case. The ICH guidelines also apply here, and it's consensus in the industry that the best way to look at this, or the most fair way to look at this, is to compare it with other chemical reactions, and more specifically to chemical catalytic reactions. Those are the closest to the use of enzymes in how to design your control strategy. But of course, if you, with that being said, you have to pay special attention to impurities that are specifically related to the use of enzymes, and also how enzymes are produced, and which impurities and risks those bring with them. So the key risks, so that being said, ICH guidelines apply. What are the key risks and factors that are associated with the use of enzymes, and that are very important to fully understand to develop your control strategy? Well, the first one and the most important one, or the most critical one, is the toxicological potential of the production strain. So all enzymes are produced in a microorganism, so a host, and that host can be either a bacteria or yeast, and that host should be class 1 biosafety 1. Well, what does that mean? That means that it's intrinsically safe, so that the microorganism, if a healthy human being is exposed to that specific microorganism, there are no adverse health effects and there's no health risks. They are generally not related to to production of, so they don't produce endotoxins, and basically most of these microorganisms are applied even in food products or as probiotics, etc. So they are safe, and the microorganism itself is intrinsically safe. The second risk factor that needs to be taken into account is related to that, and that is the impurities that are formed in the production process of the enzyme itself. So again, the enzyme is produced in a host cell, and that host cell, of course, contains DNA. It contains other proteins, host cell proteins. It can contain endotoxins, but it also contains, for example, debris from the cell walls, etc. So all these impurities can be present in your enzyme and therefore have to be taken into account in developing a control strategy. And then the third factor is the physical shape in which you use your enzyme. So there are various possibilities, all of which are applied in the industry. So you can use whole cells, you can use cell lysates, purified enzymes, or immobilized enzymes. If you use whole cells, that basically means that you use the complete bacteria or yeast in achieving your conversion. So you grow, you cultivate the organism, and then you have the living organism do the desired conversion. But this also means, of course, that you have living organisms in your reaction mixture, and that need to be killed, need to be removed from your product in your workup procedures. And it also means that all the components of the cells, so the ones mentioned in the bullet above there, are also all present in your reaction mixture and all have to be removed. A little bit better, or less risk, is using of cell lysates. So there you basically still use complete cells, but they have been killed. So the cell wall has been broken, and you have either spray dried the mix or you freeze dried it, and then you use that mix as a catalyst in your reaction. This has the benefit compared to the use of whole cells that there's no living organisms in there anymore, but you still have all the components of the original microorganism in your reaction mixture. So all the other proteins, cell wall, debris, etc., are all still in there and still need to be removed in your workup procedure, in your purification procedures. So to circumvent that, you can also use, it's better to use purified enzymes. So what you've done there, then you have produced the enzyme in the microorganism, and you kill the microorganism, you take out the enzyme, and via multiple purification step, you purify the enzyme, and then you use that purified enzyme as the catalyst in your reaction. And if you do that, then if the enzyme is 100% pure, you don't introduce any other impurities, and the only thing you have to take into account in the workup and in the analysis of your product is the potential presence of the enzyme itself and any degradation products related to the enzyme. So it could decompose, etc. So those are then still impurities that you need to take into account. And to prevent that, you can also use immobilized enzymes, so where the enzymes are connected to a bead or something like that, and then you can, after you have done the desired conversion, you can remove the enzymes that are connected to that bead by filtration, and then you basically, if you have immobilized your enzymes 100%, you can also prevent the presence of enzyme in your product relatively easily. So those first three are all related to the use specific for the use of enzymes and related to the way enzymes are produced. And the fourth and the fifth one are more general factors that need to be taken into account, but they are not really specific to the use of enzymes. So the first one is where you use where you do the biocatalytic step in the synthesis. Yeah, this is relatively easy to understand. If you do it very early, say in the first step of a long and complicated synthetic route, then after that step, you still have a lot of chemical conversions, a lot of purification steps, and the chances of anything that is related to the use of an enzyme ending up in your final product are very small or even negligible. But if you use the enzyme in the final step of your route of synthesis of your production method, then of course the chances of or the risk of something related to the enzyme ending up in your product becomes significantly higher, and you need to have better and more elaborate control strategy to control that. But this is, again, this is not related only to enzymes. The same goes if you would have used a transition metal catalyst or something like that. You would also have to do the same thing and show that no metals are present, for example. And the final one, also not strictly related to enzymes, but more general, is the route of administration. So if you have a parenteral product and you inject it directly into the bloodstream, of course, the requirements for impurities in general and also for impurities related to enzymes is much stricter and much lower levels are acceptable than if you would use if you have a tablet, for example, that is for oral administration, where the level of impurities that is allowed is much higher. So this again is general. Now in the next slide, I want to look at the focus on peptide ligase. And more specifically on omni-ligase. So as Leonard explained, we have various peptide ligases available, but omni-ligase is the one that is we often start from in our process development work, because it's a peptide ligase that is suitable for ligating many different sequences and always a good starting point. So omni-ligase is produced by in a bacteria, Bacillus subtilis, and that is a risk group 1 organism. So that means that it's a safe organism. It's not related to not connected to endotoxin production, and it's even used in probiotic products. So if you ingest this bacterium, if you're a healthy human being, ingesting this bacterium will not harm you at all. And this also means that any impurity or any side products produced in this bacterium are also not considered toxics. The expression levels of Bacillus subtilis or of the omni-ligase in this bacteria are very high, so it's in the gram per liter levels, and the largest scale so far that we have produced these enzymes at a CMO was 100 liter per batch for GMP usage. But this can be scaled up further if required, and 100 liters is roughly good for production of around 50 grams of enzyme. Very important is that omni-ligase is a purified enzyme. So after the inoculation and the production of the growing of the cell culture and the production of the enzyme is isolated as a pure enzyme through several microfiltrations. All the peptide ligases and also omni-ligases are equipped with a his tag that is used for purification. And the enzymes in the final step are also, there's also a sterile filtration. So all the enzymes are pure enzymes. And in the next slide, we have an example of a certificate of analysis of omni-ligase. and we can focus on the various examples of or the various entries in this in the certificate of analysis. So first of all, you see that the purity of the enzyme is 100%, clearly indicating that it's a purified enzyme. The enzyme is tested for the presence of endotoxins below a very low level, and it's also tested for the presence of living organisms at a very low level. Because the enzymes are purified using a HISTAG column, which is of course a nickel affinity process, the enzymes are also tested for the presence of nickel below a very low level. And then finally, there's also every enzyme batch comes with a TZ BZ statement. So the Bacillus subtilis is of of course non-animal origin or non-mammal origin, but also it's important that all the raw materials that are used to cultivate the bacteria are also BZ TZ free, and that is stated here on the certificate of analysis as well. So very important in API production is of course the cleaning of equipment. Well, for enzymes, this is not really that challenging because most the general conditions that hydrolyze the enzymes are known. So if you use aqueous basic or basic oxidizing conditions, you will normally completely hydrolyze the enzymes and hydrolyze it into the amino acid components, which are water soluble and easy to remove in cleaning. And also the testing of how clean the equipment is. You can use various techniques, but probably the easiest and recommended is total organic carbon content, which captures both the organic components and also any enzyme residues that are still there. A bit more challenging is how to ensure that there is no enzyme in your product. So ideally, for removal of the enzymes, you would want to use normal downstream processing and purification processes. And in most cases, that is sufficient to remove your enzyme. Peptide production is a little bit special because for small molecules, you can often use conditions similar to what I've shown for the cleaning. So hydrolyzing conditions to completely destroy the enzyme and then remove the amino acid components. But of course, for peptides, you cannot use those because you would also destroy your product. So you have to be a little bit more careful. But we see in our lab that on small scale, standard HPLC purifications in most cases is sufficient to remove the enzyme. But of course, this has to be established on a case-by-case basis. Each product is different and each process is different. And the closer the product gets to in properties to your enzyme, the more challenging this becomes. Then, of course, it's not only preventing the enzyme from being in your product, but you also have to have the analytical tools in place to detect if you have successfully removed the enzyme. We recommend to use ELISA using the HISTAG that all peptide ligase enzymes have. And that has the benefit that it's very sensitive, and also in this case that you even if the enzyme is partly hydrolyzed into different fragments, you would still have one fragment with a HISTAG, which would then also be the total amount of enzyme and enzyme-related fragments would be detected in this method. But other methods for detecting residual enzymes in APIs are known, like SDS-Page, and especially Merck has published quite some interesting and recent papers on this topic. One of them is mentioned here in the note, which is about SDS-Page imaging technology to detect enzymes in API. So this is the final topic of this part, and that is the optimization of enzymes. So in general, the way it normally goes is that for initial production of initial quantities of an NCE or a candidate, enzymes that are commercially available from kits or whatever are used, and those can then be used for up to and including phase 1, assuming that you can develop a decent process using this enzyme. And then around phase 1 or going into phase 2, then you can optimize the enzyme, and that's also the phase where it makes sense to possibly optimize your enzyme. So it's not a requirement. If you are happy with how the process is performing and the enzyme is performing very well, you can also, of course, just stick to the enzyme. But if your quantities of product you need to produce are increasing and cost of goods become more important, and also the chances of the product making it to market increase, companies get interested in optimizing the production ways, and then it makes sense to optimize the enzyme to get higher turnover numbers and more efficient or better cost of goods. And this is still allowed. So regulatory authorities are allowing modifications and optimization of the enzyme all the way up to phase 3. Of course, this needs to be carefully explained, and a good file needs to be built, but this is possible. Connecting this to our peptide ligases, this would also typically be what we recommend. So again, we would start with omni-ligase, although we also have other enzymes off the shelf available that we can also start with. And then if your product reaches phase 2, we can do enzyme screening and enzyme optimization to improve turnover numbers, improve cost of goods, etc. And for people that are interested in this, Leonard recorded a webinar on this topic a while back, also a Bachem webinar, so you can still see that webinar you can find it on the Bachem website. And then, this is the last slide. So summary. So enzyme, the CEPS technology, the chemo-enzymatic peptide synthesis, allows the making of peptides that cannot be efficiently manufactured with solid phase peptide synthesis. And these are mainly the longer peptides, synthetic proteins, and also the cyclic peptides, which are very good. The technology works very well to make cyclic peptides as well. It's sustainable, it's cost efficient compared to chemical methods. The chemo-enzymatic peptide synthesis can be done on commercial scale. So the enzymes are produced on commercial scale, and also the processes using the enzymes can be done on large scale. Around the use of enzymes in general and peptide ligase specifically, a case-by-case risk-based control strategy is required to use the enzymes, and I've highlighted some of the key considerations there and how to, or at least the pillars on which to base this control, risk-based control strategy. And then finally, EnzyTag and Bachem have a partnership. So if you are interested in having your peptide NCE produced using the peptide ligase technology, thank you.
Great. So thank you, Robert, and thank you, Leonard, for a very interesting and deep insight into this technology and related aspects. Yes, we are now live on stage. This is now the live Q&A session here. And before we start with this, we are always curious about some answers from your side regarding this CEPS technology. And our question of today's webinar is, what is your main concern using an enzyme for peptide synthesis? So on your screen, you should now have a pop-up window where you have multiple choice options. Please feel free to choose your favorite, and I will let you know the results in some seconds. Okay, we have a winner, but not a full clear winner. 45% have a concern, and that is the favorite for enzyme removal and detection of the enzyme in the API, followed by 2 33% answers with the enzyme production itself, and maybe potential hurdles and concerns with regulatory guidelines. So thanks a lot for that. That's very helpful for EnzyTag and also for Bachem to further pursue that. And yes, we will now open the live Q&A session. And let's see what comes in. Okay, already detected the first question. The question is, does different peptides require different enzymes? So Robert and Lennart, could you comment on that?
Yeah, thanks for the question. The answer is yes. But also no. So let me first start with the no. The enzyme that Robert Maas presented is a broad spectrum enzyme. So it is designed to recognize the broadest possible panel of peptides to ligate together. Of course, you're sacrificing on something, and that is that you will never get complete or 100% conversion. And so in order to reach that, you can do enzyme engineering to make it specific for the ligation site you've selected with the omniligase enzyme. So that's also typically how we do it. We start with omniligase, and then along the clinical development trajectory of the drug candidate, we do engineering of the enzyme in order to improve the conversion.
Yeah, great. Thanks a lot. Next question is, can these enzymes be produced in E. coli or E. coli?
I think with a lot of engineering might be possible, but B. subtilis is a better organism to do so.
Okay, good. Then the next question is, you said that it is not necessary to use protection groups. Is it possible to synthesize peptides from simple amino acids? Example, coupling of valine with lysine?
I can also take this one then. Yeah, so then you're essentially using it as a alternative or a substitute for a coupling reagent. And so the enzyme requires 6 amino acids in the recognition pocket. So coupling single amino acids together with the enzyme is hardly possible. So the enzyme is not active enough to realize that. And certainly the traditional peptide coupling reagents beat the peptiligase enzyme there. You need to have a certain length.
Yes, you mentioned the former webinars, it's 6 amino acids, which are in the pocket of the enzymes. Great. Then the next question is, what is the smallest and largest peptide fragment size that can be ligated together with the enzyme?
Yeah, so maybe I can answer that one then. It's related to the previous question. So the smallest is 4+2 basically, and the largest is, yeah, there's not really a limit regarding the largest. It's more related to what makes sense economically, how large you want to make the fragments. Can be from 20 to, but we did 60, 70, those kind of, or you can ligate to proteins even. So there's no size limit, no upper limit.
Yeah, okay. And maybe in that context, another question is, what's the longest peptide being synthesized with peptide ligase so far?
The largest we actually made, yeah, we did up to 110 Leonard, but yeah, I think that's the largest that we did in a commercial project. But there's no, I mean, that's just the largest we did, I think, and that was a 2-fragment strategy, but longer is possible. Also, if you use multiple fragments, protected fragments, where you deep protect and then couple the next fragment, you can go higher. We mainly run into the ligations work. The problems sometimes arise from solubility and those kind of things. That's the biggest challenge when you go that far.
Okay, great, thank you. A further question is, do you immobilize your ligases for peptide production?
It is possible. So we don't do it on a routine basis, but we have tried it in a research environment. So at the other end of the enzyme, so to speak, you can attach it to a bead, and then you can just flow a solution of a peptide solution over the beads, and then you get conversion. You can even recover the beads with the active enzyme and do that another time and another time. And I think we've shown up to 5 times that we can do it. That was in a simple R&D environment, but for sure, if you put a bit more development into this, it will certainly be possible.
Great, thank you. Further question is, can the ligase work on unnatural amino acids in the peptide, even at the end?
Well, it depends on the natural amino acids, and it depends on the position. So we know that in some specific pockets it works, but in general, yeah, it works better if you keep the unnatural amino acids outside of the recognition pockets. That's the safest answer.
Okay. Wow, so many questions. Great. Another question is, do we need to make antibodies against omni-ligase for detection in ELISA tests?
Yeah, well, the answer is no, because of the, like I mentioned in the webinar, that all the peptide ligases have a his tag. You can get ELISA, or you can get these ELISA kits that are specific for the his tag, so you can use that.
Okay. Good. And then a further question is coming in, saying, what's most advanced clinical development stage and batch scale example of released API amounts? So, yeah, the question, are these is this technology already in clinical stages and maybe already available commercially?
It's not yet in a clinical stage, but we are working on this in projects to get to that point. The batch scale, so if I interpret that correctly, is then it has been proven at slightly less than kg scale in a GMP environment. And that means that the fragments have been ligated together with the enzyme at a scale which is just below a kg, and that API has been released. So another one is that the first DMF has been filed not so long ago, and that has been published also not so long ago. So there are quite a bit of companies working with this technology and maturing it.
Great. Okay. I think we covered nearly all questions. Thanks a lot for your great interest. So just to let you know, at the beginning, I said this is already the 4th webinar about the SEPS technology. If you're interested in the former ones, please go to our website here or our Bachem YouTube channel to find them. There's also an upcoming talk about the SEPS technology that's in a conference in Reston, Virginia, with Leonard and with Stefan Eisler from Bachem about the comparison of SPPS, native chemical ligation, and SEPS technology. And as the last one is, yeah, we have also updated our company brochure. That's also maybe worth looking in. Yes, and you will all get the recording pretty soon sent by email to you. And with that, I thank you very much for your attention. It was a great pleasure to have you all here, also with your interesting questions. Thanks a lot, Leonard. Thanks a lot, Robert. Yeah, I'm looking forward to hearing more about this amazing technology. And with that, I'm saying goodbye. I'm wishing you a nice rest of the day, be it the morning, the afternoon, or the evening. Yes, and I hope to see and hear you soon. Goodbye. All the best. Take care.